Hey folks, Arjun here with another dose of sweet, juicy endocrine with all the glands you could possibly want. As with our other videos, we here at First Aid Express like to ask questions, and it's not because we like to hear ourselves talk. Even though you could wait the 1 or 2 seconds it takes to hear the answer, I strongly encourage you to try to come up with the answer on your own. It'll help keep you engaged and give you sort of an idea of how well you actually know some of this stuff. Today we'll be talking about physiology, which is the real meat and potatoes of endocrinology. So pop those eyelids open, and let's start off with one of the highest yield points, glucose regulation. Our discussion, of course, begins with insulin, secreted by the pancreatic beta cells. Now, before starting with the nitty-gritty biochemical mechanisms, I just want to go over what I consider to be the core of endocrinology and the highest yield step 1 stuff, the cause and effect of homeostasis. So to really generalize, what's the purpose of insulin? The purpose, quote unquote, is to coordinate the body's resources during times when glucose is plentiful, primarily through anabolism, or building up complex molecules. Well, how does it do that? The most intuitive way the body deals with a surplus of glucose is to store it, and that job goes primarily to our 3 reliable friends, the skeletal muscle, liver, and adipose tissue. When glucose is plentiful, insulin causes skeletal muscle to import and metabolize glucose, storing it as glycogen. The process is similar for adipocytes, but logically, fatty acid and triglyceride synthesis is the primary means of storage. The liver's a little different. Insulin actually has no power to control the transport of glucose into the liver, but it does cause increased glucose metabolism. We'll talk about exactly why that is in just a moment. So which does the liver preferentially synthesize, glycogen or fatty acids? Preferentially, the answer is glycogen, but the liver's glycogen tanks fill up pretty quickly. When the storage capacity has been reached, fatty acid synthesis becomes pretty important, because the liver can always count on the adipocytes to help with the storage. Remember, the net adipocyte capacity is way bigger than the liver's capacity to store energy. The liver just has to package the fatty acids up as triglycerides, send them off with a VLDL postmark, and away they go. FYI, if you think the adipocytes are also letting any of that sweet fatty goodness go, you got another thing coming. Insulin also inhibits lipolysis in the adipocytes. Finally, if insulin is an anabolic hormone, what would you expect its effects to be on protein metabolism? Insulin causes increased amino acid uptake and protein synthesis in all cells while inhibiting proteolysis, which makes sense since protein synthesis takes a ton of ATP, and that's only really found when macronutrients are plentiful. And finally, for real, insulin increases the activity of the sodium-potassium ATPase pump. This might seem pretty trivial now, but it becomes pretty important in the treatment of hyperkalemia. Check this out in the renal section on potassium regulation. So hopefully that was a helpful demonstration of the big picture of insulin. Now I'm going to get into the mechanisms of how these regulations take place. Let's start with the secretion of insulin itself. This humble rectangle represents the beta cell, the place where it all begins. Now before glucose even enters the picture, this tiny warrior cranks out granules of stored insulin day and night. The hormone is actually synthesized as a prohormone and packed into vesicles. And this, my friends, is where the magic happens. Because inside the vesicles, proinsulin is cleaved to form active insulin and the inactive C-peptide. And these granules full of insulin and C-peptide park themselves right by the cell membrane, ready to be secreted at the drop of a hat. So the question of the day is, as always, why does anyone care? The real clinical value, and the one you'll be asked about on step 1, is the ability to distinguish between different etiologies of hyperinsulinemia. Think about it. If you've got an insulin-secreting tumor, otherwise known as an insulinoma, you'll have high levels of both insulin and C-peptide, because they're synthesized together by the beta cells. What if you've taken a mega overdose of regular insulin? I don't think so. Endogenous insulin is always accompanied by the co-release of C-peptide, but exogenous insulin never is. So how do you get from high glucose to insulin release? It's a bit of a complicated song and dance that unfortunately you have to memorize for this test, but it's actually quite a bit easier when you see it animated than it is on paper. More glucose on the outside of the cell means more glucose transport into the beta cell, where it gets catabolized to generate ATP. What follows is an inhibitory step where ATP actually deactivates the ATP-sensitive potassium channel. Without the leak channels working, positive charge builds up on the inside of the cell because potassium cannot escape. This depolarizes the beta cells, which leads to the next step. Now this inhibitory step is always the hardest for me to remember, but if you remember that the goal of this exercise is to inhibit the potassium channel, you should be golden. The depolarization of the cell opens everyone's favorite vesicle releasing channel, the voltage-gated calcium channel. Just like at the axon terminal, this allows calcium to enter the cell and permit vesicle release. Now that we've taken a look at insulin release and regulation, let's see what it does once it's at its target. Insulin's actions are mediated via the intrinsic tyrosine kinase or MAP kinase pathway. It's one of the more esoteric pathways, so I'm going to quickly take you through how it works. Insulin binds to the intrinsic tyrosine kinase receptor, giving each subunit the ability to add phosphate groups to the other. Once autophosphorylation takes place, a whole host of adapter proteins glom onto the activated receptor, the final result being the activation of two proteins, phosphoinositol 3 kinase, or PI3K, that acts in the cytoplasm, and MAP kinase, which translocates to the nucleus to control gene expression. Now, what does the signal cascade accomplish? As a matter of fact, everything you see here. So now we're going to get into the different cell-specific effects of insulin and how these different types of cells respond. We'll start off with glucose transport. So remember how I said that insulin facilitates glucose transport in two of these but not the third? Both myocytes and adipocytes possess vesicles containing the GLUT4 transporter, and insulin's PI3K pathway causes vesicles to integrate with the cell membrane, increasing the cell's ability to import glucose. In the case of striated muscle, the GLUT4 transporters can also be translocated to the membrane with exercise, which explains why exercise is so important for insulin-resistant diabetics. Can you think of a way this might go wrong? I know, you're probably not used to thinking of exercise as a harmful thing, but this is actually a pretty common cause of hypoglycemia that you might see on step 1. Patients who take insulin are supposed to decrease their insulin dose before exercising to account for the dual drop in blood sugar, but when they forget, it can lead to some pretty significant hypoglycemia. So how does the liver get its glucose? It uses the GLUT2 transporter, a transporter that isn't controlled by insulin. Now, I know it's weird to spend so much time on a transporter that's not insulin dependent on the insulin chapter, but this transporter is actually pretty unique, so bear with me. The first cool thing about it is that it's bidirectional. Glucose can either enter or exit the cell. Why is that so important for the liver? Because unlike other cells, the liver doesn't just want to suck up all the nutrients it can like a fat pig. It needs to be able to secrete glucose into the bloodstream when the body's in the fasting state. You can see that I drew GLUT2 long and skinny compared to the big chubby GLUT4. Now that's not by accident. At lower and most physiologic glucose concentrations, the liver transports glucose slowly when compared to GLUT4. It lets the other tissues have first dibs when supplies are low. But it also has an insulin-independent mechanism for soaking up glucose when levels are too high. As you can see in the diagram above, the GLUT4 transporter maxes out pretty quickly, whereas the GLUT2 transporter has a high capacity for transporting large amounts of glucose. To use the terms you use in pharmacology, GLUT4 has the lower Km, but GLUT2 has the higher Vmax. Anyone have a feeling of biochemical deja vu at this point? Or have you seen this comparison before? There's actually a similar comparison, weirdly similar in fact, to the first step of glycolysis, catalyzed by glucokinase in the liver and hexokinase just about everywhere else. Glucokinase, like GLUT2, has a high capacity that allows it to chew up a lot of glucose at high concentrations, but a low affinity that leaves glucose alone at low concentrations. The big difference? In this case, it's the liver's isomer, glucokinase, that's regulated by insulin. Finally, those of you observant folks out there may remember that the glucose transporter on the beta cell from a couple slides ago was also labeled GLUT2. Now, why do you suppose it's important that these cells have a high-capacity transporter? Because they need to know that there's a lot of glucose so they can secrete a correspondingly large amount of insulin. Think about it. If they had a low-capacity transporter, like GLUT4, they'd increase their levels up to about 120, and then past that point, they wouldn't realize that any more glucose existed because the rate of glucose transported would be the same. They'd be all, well, I think I'm about done here, and not change their insulin secretion, and that'd be bad news bears. We want our beta cells to be able to modulate insulin levels over an extremely wide variety of glucose concentrations, like GLUT2. There are a few other important glucose transporters, none of which is regulated by insulin. For starters, these two behemoths, GLUT1 and GLUT2, are very high affinity, high capacity transporters. GLUT1 is mostly in red blood cells and the blood-brain barrier, while GLUT3 is the main transporter in neurons. Now, carrying high levels of either of these transporters is like having access to an all-you-can-eat buffet of glucose, even in times of starvation when insulin is scarce. To be fair, I think we can afford the freaking brain that luxury, but what about the red blood cells? Why do they get all the glucose? Remember, RBCs have no mitochondria and can't undergo anything more efficient than anaerobic metabolism, so they need glucose to meet their energy requirements. They are physically incapable of oxidizing fats. Now this weirdo down here is called GLUT5, and contrary to what its name sounds like, it's actually a fructose transporter. There aren't too many cells that use fructose, but the small intestine can absorb it and convert it to glucose for the rest of the body, and sperm actually use it for energy. Now once you know insulin, glucagon's not really too hard to get the hang of. In many ways, it's kind of like the anti-insulin. Insulin is secreted by beta cells in response to hyperglycemia, whereas glucagon, well, I'll let you do this one for me. What's glucagon secreted by? The alpha cells, right? In response to hypoglycemia. Insulin operates via the intrinsic Y kinase pathway, whereas glucagon is just your average Y-bread G protein coupled stimulatory pathway. Insulin lowers the blood sugar and promotes anabolism by inducing glycolysis, promoting glycogen storage, and by promoting fatty acid and triglyceride synthesis. So what does glucagon do for the metabolism? It raises the blood sugar and promotes catabolism by inducing gluconeogenesis, glycolysis, lipolysis, and ketogenesis. So that's the easy part. What's harder is knowing the deviations from the opposite game I just played in front of you. First of all, insulin deals more directly with protein anabolism, whereas glucagon, as far as we know, doesn't have any direct effects on protein metabolism. Glucagon also doesn't have any effects on glucose transportation, whereas insulin upregulates it on certain cells. And finally, insulin kicks glucagon's butt any day of the week. It can directly prevent the alpha cells from producing glucagon. And I think that about covers it for these two hormones. There's actually a lot more important stuff regarding the effects of insulin and glucagon on specific metabolic pathways, but that's covered in way more detail in the metabolism section of biochemistry. Your job, when you're done listening to me, is to go review that section of biochemistry and see where insulin and glucagon regulate carbohydrate metabolism, because now you'll be reading it with a sense of perspective. But for now, in the endocrine section, we're done with insulin and glucagon, and it's time for a flash quiz. We'll keep this quick. Which glucose transporter is slowest at low glucose concentrations? Go ahead, pause the video if you need to think. And the answer is the GLUT2 transporter, found both on the liver and the pancreatic beta cells. Now it's time to hit up the main body of endocrinology, the hypothalamic pituitary axis.